This video talks about the calcium binding in skeletal muscle, cardiac muscle, and smooth muscle, and how they differ from one another, or how they're similar to one another. But before we move on to the binding of calcium, let's take a quick review of how calcium binding, or how muscles really work in its most basic unit. So imagine that this is a muscle right here. The red structure right here is a muscle, and this yellow structure that we see here is a nerve which is coming to the muscles and stimulating the muscle. A nerve should be much smaller than the muscle itself, but this is just a diagrammatic picture, not very accurate in terms of proportions. So let's just keep that in mind. Imagine this is the nerve terminal coming and stimulating the muscle right here. And this arrow here means that we are taking a closer look, a magnified look of what's happening exactly inside this muscle. So let's take a quick look what happens. When we take a quick look and when we take a small section of that skeletal muscle, we're going to see that the myofibrils kind of run parallel to each other, like so, the way it's shown in this particular diagram. Now these are all packed into one structure with the sarcolemma and endoplasmic reticulum. Again, we're going to take a quick peek even at a more magnified view in this picture. So when we take a look at the myofibril right here, which was one unit inside that packed structure, we see that there is the thick bands and the thin bands in between, the thick and the thin bands, and these kind of units are called sarcomere, and I'm sure you know that by now. And these structures fulfills the myofibril. By the way, myofibril is the basic unit of muscle. Just like cell is a basic unit of life, myofibril is a basic unit of muscle. So again, this is a more magnified version of a sarcomere. The black lines are the thick bands or the myosin bands. The purple line are the thin bands or the actin bands. This is A band. A band consists of only the thick filaments. You see, even though it has overlap of the actin right here and overlap between the actin and the myosin, when we talk about the A band, we are only talking about how thick the thick band is. We don't care if there is overlap at all. So from here to here is our, sorry, my drawing ability is really bad. From here to here is the thick band or the A band, and the region where we have so this is band, by the way, don't confuse it with anything else. It's A band. And A band never changes its shape, and it can't really. I mean, how can you change the shape of the thick filament? It's not changing with the cross bridging, it just stays the way it is. And the purple region where there is no region of overlap is called the I band. Okay, so this is called the I band. Now, like I said, this is the thick band region, is the A band. The thin band with no overlaps from the thick bands is called the I band. And this area, which has no overlap of thick bands, it's only thick bands from here to here. This region is called the H zone, not H band, but H zone. So just wanted to throw it out there for a quick review. Okay, let's move on. So again, this is a picture of myofibrils running vertically inside this sarcolemma. So these are the myofibrils, and this is the sarcolemma. Sarcolemma is the plasma membrane of a muscle, and these green structures is the sarcoplasmic reticulum. Sarcoplasmic reticulum it's like a hairband which kind of encompasses all the myofibrils which is running along. And often we are going to see T-tubules running inside like so from the sarcolemma inside the myofibrils, and which not only increases surface area, but it also triggers the action potential to kind of go inside the myofibrils more thoroughly. So moving on, just wanted to mention that when we're talking about cardiac muscle and skeletal muscle, a big difference from cardiac muscle to skeletal muscle is that cardiac muscle gets its calcium from the plasma, where the calcium in the skeletal muscle is actually coming from inside the T-tubules. It's not actually coming from the plasma, so that's an important distinction that we have to remember, that the calcium from the plasma stimulates the cardiac muscle, which is not the case for skeletal muscle, because for skeletal muscle it's the T-tubules which kind of stores all the calcium. So moving on, again this is the sarcolemma invaginating, forming the T-tubule, and then in these T-tubules there is these receptors, these green bubbles are called the dihydropyridine receptors. Dihydropyridine receptors are calcium sensing receptors. Okay, they are going to release calcium onto the T-tubules. Now this is a picture of how the cross bridging happens. Let me orient you really quickly. This block right here is the actin. This block here is the troponin. Actin is a part of the thin filament. This is tropomyosin until here. This is the tropomyosin. This light purple band is a tropomyosin, and this red box right here is troponin. When we're talking about thin filaments include actin, thin filaments include tropomyosin, and thin filaments include troponin. All these are going to make the thin filaments. The only thing that makes the thick filament is going to be troponin. Now, another naming, let's get the naming out of the way. This pit right here is called the attachment site for the myosin heads, and these pits that are coming out, these are called the cross bridges. So let's take a quick look at that. So what happens is, let me go back to the other picture. So what happens is whenever there is a propagation of signal coming down the sarcolemma inside the T-tubules, like it's shown in this picture. So imagine this uh, green line that's coming down. The green squiggly line is the wave of depolarization. It's kind of stimulating the dihydropyridine receptors and causing the action potential to kind of stimulate all the dihydropyridine receptors. This releases calcium. And how does it release calcium? So the green line, the action potential, is stimulating the dihydropyridine receptor. Now the dihydropyridine receptor is in turn attached to something called the ryanodine channels. So when the ryanodine channels get stimulated through the dihydropyridine receptors, it releases calcium into the system. So here you can see how the calcium is being released into the system, and the calcium now leaks back into the T-tubules, and this is going to go and bind to troponin to have the actin and the myosin bind together. So let's go back to that picture. So now here we see that the calcium is going and binding to the red box, which is the troponin. The blue line right here is the tropomyosin. And as soon as this happens, as soon as calcium binds to the troponin, it starts pulling on to the troponin and leaving the actin and the myosin to bind together. So going back to this picture, we can see that the calcium from the sarcolemma, from the T-tubules, the calcium comes and binds onto the pit of the troponin right here. So once it binds in the troponin block right here, this tropomyosin is going to get removed. So it's going to start moving away, something like this. As you can see, the tropomyosin is moving away as calcium binds to it, leaving the cross bridging from happening so much easier because the attachment sites are there, the pits are here, they can go and form cross bridges. So once they form cross bridges, that's what they look like, and you can see the tropomyosin is kind of out of the way. And now that the cross bridging happened here, for this cross bridging to kind of break apart, we need an ATPase. So this is where I'm showing that it has to break apart, and we need an enzyme called cercoplasmic ATPase. And when the cercoplasmic ATPase is going to come, it's going to release that calcium from the troponin, and the calcium is going to go back to the T tubules. It's going to go back to the ryanodine channels, and it's just going to get stored there for next time it needs to be used. So that is the quick review of exactly how cross bridging happens during skeletal muscle or cardiac muscle contraction. Now, it's very, very similar for skeletal and cardiac muscle contraction. The only difference is when it comes to smooth muscle. For smooth muscle, instead of binding to troponin, they're going to bind to something called calmodulin. So that is one specific difference that we have to know. So for skeletal and cardiac muscle is going to be troponin, even though the structure itself from troponin is a little different for cardiac and smooth muscle, but that is insignificant in terms of USMLE step 1. The more important fact that it is going to be the same in terms of mechanism, but it's going to be different in terms of instead of troponin, we are going to have calmodulin in smooth muscle. One more thing I wanted to kind of throw it out there is that there is 3 different types of troponin. There is the troponin C, which binds calcium, the troponin T, which binds to the tropomyosin, forming the troponin and tropomyosin complex very strong, and troponin I is the one that binds to actin, again making the whole complex really, really strong. So let's take a quick look. So this is the binding of troponin T with troponin and tropomyosin, and this is the binding of troponin with actin, making this whole structure very, very solid and very, very so that it doesn't break very easily. Now let's look at our example that we talked about, that we're going to talk about at the very beginning. In our example, it says that in skeletal, cardiac, and I put down in brackets smooth muscle because I wanted you to think about smooth muscle as we're talking about skeletal and cardiac. Binding of calcium to which of the following proteins is most likely to be associated with active force? In this case, the answer is going to be troponin, as simple as that. And for smooth muscle, it's going to be calmodulin.